A cell wall is a structural layer surrounding plant cells just outside the cell membrane. It means in animals there is no cell wall, but in plants the outermost layer is cell wall, which is made up of polysaccharides. These polysaccharides are cellulose. The cell wall was discovered by Robert Hooke in 1665. It was discovered earlier than protoplast. It is the outermost boundary of the plant cells. It means the plant cells contain cell wall, then cell membrane. It is secreted by protoplasm of the cell, the organelle, which is known as Golgi bodies. The Golgi bodies secrete polysaccharides, and as cell wall is mostly polysaccharides, it is secreted by Golgi bodies. Students, you should know that Golgi body secrete polysaccharide, while disaccharides are synthesized by endoplasmic reticulum, smooth endoplasmic reticulum. Its thickness varies in different cells of the plant, as the younger cell will contain thinner cell wall, while the old or oldest one will contain thickest cell wall. So thickness varies depending upon the age of the cell. Each cell, whether isolated or occurring in tissues, has its own cell wall. The first formed cell wall is known as primary cell wall. It is present in the meristematic tissues. So when a cell is formed, it contains its own cell wall. You should remember this. Now the next point is each cell wall of neighboring cell are held together by an intercellular substance called middle lamella. Each wall varies greatly in morphology and chemical composition, but its characteristics have a close relation with the age and function of the cell. So there will be meristematic, parenchyma, collenchyma and sclerenchyma depending upon the nature of cell wall. Fundamental parts of cell wall. The cell walls of plant cells have three fundamental parts. The names of these parts are middle lamella, primary cell wall and secondary cell wall. The composition of these three are different and the functions are different as well. We will learn that middle lamella, primary cell wall and secondary cell wall are present in different cells. Now connecting to the point that middle lamella cement together the cell walls of the neighboring cells. Middle lamella contain compounds which are known as pectins. These pectins are cementing in nature. It is usually thin and about 1 micron in thickness. The middle lamella is the thinnest part in the cell wall. In woody tissues, the middle lamella is commonly lignified. It means it contains lignine. Lignine is a special type of heteropolysaccharide. We will learn the nature in this lecture as well. It is also known as wood fiber. Primary cell wall. Primary wall is the first wall formed in a developing cell. It means when the meristematic tissues form new cells, the new formed cells contain primary cell wall. It is usually 1 to 3 micron in thickness. 1 to 3 micron in thickness means 1 micron is the millionth part of a meter. So it is 1 to 3 micron in thickness. It is elastic, it grows with the cell, extendable, crystalline, and optically active. It is crystallized and optically active means it bends the polarized light. When it passes through, it bends the light. So the compound which show optical activity are known as optically active compounds. Optical active compounds is capable of rotating the plane of vibration of polarized light to the left or right. When it is rotated to the right, it is known as dextrorotatory. When it rotates to the left, it is known as levorotatory. So the primary cell wall contain fibers, cellulose fibers and matrix. Inside the cellulose fibers, the pectic compounds are present. It is composed of cellulose pectic compounds. From Greek pectikos means coagulating. These hold together the fibrils of cellulose, while pectin is a structural acidic heteropolysaccharide contained in the primary wall and middle lamella of terrestrial plants. So in short, pectin hold the cellulose fibrils together and make it stronger. You should also remember this, that cell wall is hyaline, glass-like when it is wet. Its main component is a sugar acid derived from galactose. We are talking about pectin. The main component of pectin is a sugar acid derived from galactose, and the heteropolysaccharide which is formed and hold cells together is known as pectin. I was also talking about the nature of cell wall. So when it is wet, it is clear, so the photosynthetic pigment receives much light by this way. It also contains polysaccharides like Hemi Cellulose and other polysaccharides. The cell wall is a unique structure which is present in the plant cells, and it contains polysaccharides and hemicellulose. Hemicellulose is a polysaccharide, heteropolysaccharide, which contain xylose, mannose and other units. It contains cis bonding like cellulose, but the unit is not glucose. That's why it is known as hemicellulose. It is present 30 to 35% in plant cells. Hemicellulose is present in the matrix of the cell wall. It is present in amorphous shape because there is no shape, so it is known as amorphous. Secondary cell wall. The secondary cell wall follows the order of primary wall in development. It means that it is formed after the primary cell wall and deposited on the inside of the cell wall. It is laid down inside of the primary wall, and the deposition is different in different cells. For example, in colon chyma, the deposition of secondary cell wall is partial. not uniform, but in sclerenchyma the deposition is uniform, as well as the thickness is different, but normally its thickness is about 5 to 10 micron in meter, more or less rigid, crystalline, and strongly optically active. It shows high optical activity. It bends the polarized light more. Secondary cell wall is also different in composition when in collenchyma and sclerenchyma, but generally it is mainly composed of lignin, cellulose, and non-cellulosic polysaccharide, Hemi cellulose, and mineral salts like calcium, magnesium, potassium, and some silica. In collenchyma and sclerenchyma, the main difference of the secondary cell wall is that the secondary cell wall of collenchyma does not possess lignin. Lignin is a heteropolysaccharide which contains alcohol groups in it. There are three types of plants regarding the nature of lignin. For example, grasses contain no alcohol group. The conifers, coniferal lignin that contain one alcohol group, and syringeal or the plants like the dicot, they contain two alcohol groups. As you can see in the diagram, middle lamella is outermost, then primary cell wall, and then the secondary cell wall. Lignin is a complex organic polymer deposited in the cell walls of many plants, making them rigid and woody. There are three types of lignin. These types are present in grasses, which contain no alcohol groups, or these are present in conifers, which contain one alcohol group, and the third one is present in the hard dicots, which contain two alcohol groups. So lignin is a heteropolysaccharide. It contains more than one types of sugar units and some other compounds, which make it very strong. By this way, plants can withstand harsh conditions and they can grow taller and lift up a high weight of leaves and branches. Lignin is a wood fiber. So we can say that lignin is present in the sclerenchyma cells. Sclerenchyma cells are those cells which are stone-like or hard cells in plants. Later on we will learn. A major role of cell wall is to protect cells from overexpansion. It means it is like a tire protecting a tube inside it. The protoplast, cell membrane, and cytoplasms are very delicate. When cell wall is there, its protection is guaranteed. So a major role of the cell wall is to protect cells from overexpansion. The cell wall on one side gives elasticity to the cell, which allows it to grow. So on one hand the cell wall protects the cell, on another hand it allows it to grow and to regain its size, but also protect it from overexpansions. Plus the cell wall maintains the shape of the cell. There some MCQs will come regarding cell wall. For example, apoplast movement. The movement through cell wall is known as apoplast. Another MCQ, that cell wall is secreted by Golgi bodies. Polysaccharides are secreted by Golgi bodies. The third MCQ it may come is it is secreted by protoplast. The fourth one may come in lecturership exams as well as in ETA exams, that the cell wall of sclerenchyma contain lignin, while the secondary cell wall of collenchyma is without lignin. Thank you.